Okay, good afternoon, everyone. On behalf of Soeres, I would like to welcome you to this fourth webinar session in a series of 9. Happy to see so many familiar names, not faces, because most cameras are uh, shut down, which is fine. Just keep it that way. I'm very happy to have this webinars, and we can only do that in a very strong collaboration with Vetworks and Poultry Health Sciences. And today, for the fourth, for number 4 uh, webinar session, we have a session on the impact of mycotoxins on poultry health and food safety. And we have a very special guest today, and that's Professor Gunther Antonissen. He's the shareholder of the chair Poultry Health Sciences of the University of Kent, and he is an expert in intestinal poultry health and also, of course, in mycotoxins and feed additives. I'm going to leave the floor to you, Professor. I think you will introduce the share as well to the people. I would say enjoy this session. Please come back tomorrow as well, and the day after tomorrow also. So if you have some questions, we prefer you to drop them in the Q&A, and at the end we will try to bundle them all on one or on a few slides. Just to come back to the previous sessions before I forget, we will all send you a link where you can access the previous trainings, and we will add these ones of today and the upcoming days as well. Please, Professor, the floor is yours. Thank you.
Thank you, Eric, for this nice introduction. To start the presentation, I will start sharing my screen. So good afternoon, everyone. So I'm Gunther Antonissen, working at Ghent University. And today we're going to talk about the impact of mycotoxins on poultry health and food safety. In the coming days, tomorrow, we're also going to discuss about occurrence of mycotoxins, and we will focus on sub-Saharan Africa for that. And the day after tomorrow, we will talk about mitigation strategies. How can we use, for example, feed additives to tackle this problem of mycotoxins? But let's start with the introducing also the chair of poultry health science. As Eric mentioned already, I'm the chairholder of this chair. This is actually a collaboration between Ghent University, the companies Poulfarm and Vetworks. And it's a partnership which we want to encourage the international collaboration from one side for research, but also for the aspect of education. For education, we are currently developing an international postgraduate which will start next from next year onwards, from the academic year from September 2021. And in the coming years, we will also start this not only in Belgium, but we will also start the course in different regions in the world. Or if we talk about research in this chair, actually it's quite diverse. Gut health is one of the major aspects, but definitely also mycotoxins is one of them. But currently we're also looking to projects looking to animal welfare, looking to parasites, so it's getting very diverse. But let me also introduce you to our mycotoxin research at Ghent University. So at Ghent University, different research group. I'm from the Faculty of Veterinary Medicine, but also colleagues from the Faculty of Pharmaceutical Sciences, Bioscience Engineering. We have a kind of collaboration. It's called MyTox, and some of our colleagues are studying the impact of mycotoxins on human health. We are studying it on veterinary health, but we've got even colleagues looking to the mycotoxin production, the fungal growth, really at the feet. So it's quite diverse, our expertise. And a couple of years ago, actually, what we did, we established myTox South. This is a kind of organization. It's a joint venture between us at Ghent University with many different partners in the South, where we try to collaborate in research project proposals in the theme, of course, of mycotoxins, but definitely for subjects which are important for the South. For example, now we've got a research collaboration, it's called MycoSafe South, where we also focus on the impact of mycotoxins, more specifically aflatoxins and fumonigens, which we won't talk today much more on that, the impact on poultry health and how can we use mycotoxin binders for this. A last introduction slide on our department. So our Department of Toxicology, we are doing quite a lot of research on mycotoxins. If you look to the pictures, you can see we are working. Just going to use a pointer. So we are looking to many different animal species, but we are mostly focusing on poultry, of course. So we are looking to what's the toxicity of these substances, but also to new mycotoxins, because every year, let's say, we find some new ones, but also to the interaction between different infectious diseases. Are mycotoxins making the animals much more sensitive to get a kind of infectious diseases, for example, Clostridium perfringens, necrotic enteritis. So we will talk on that also today. So I would suggest let's jump into the presentation and let me introduce you to the topic of mycotoxins. So mycotoxins are secondary toxic compounds which are produced by different types of fungi, meaning that this can be either Aspergillus, Penicillium, and Fusarium. There's of course already the question, why are these fungi producing them? Why are they, does they have any benefit? Yes, these fungi got a kind of benefit of producing these toxins in acquiring their nutrients to grow. So if they produce a certain toxin, they can, for example, win the game against other fungi, bacteria, to get nutrients. And actually, a very long time ago, the first mycotoxin discovered is one of our major antibiotics, penicillin. So not every mycotoxin, let's call it, is a bad one. But if we use the definition, or if we use the term mycotoxin, then we really are talking about the one which are inducing harmful effects on animal health, on human health. So today we will not talk about new antibiotics. We really talk about these fungal metabolites which are toxic for the animal. The field of mycotoxins is very diverse. A few mycotoxins we know already for many years, like aflatoxin was detected in the 1960s. But if we look now, we know more than 700 different mycotoxins, different metabolites, and they are all quite diverse. Here you can see some chemical structures and of the major mycotoxins, and none of them look the same. And it's this chemical structure which in the end is responsible for the toxic effect. And it's also this chemical structure which is almost impossible to damage, to get rid of this structure. So it's almost, when you cook the food, when you cook, when you store it, when you process it. So it's very hard to get rid of the toxicity of these mycotoxins. That's also the reason why they are such a problem. So it doesn't matter if you process the feed or you cook the feed. And if you heat it until very high temperature, it doesn't matter, they will be stable, so the toxicity will remain. Then if we look specifically to your region, Sub-Saharan Africa, which mycotoxin, we will talk much more on that tomorrow. But in Sub-Saharan Africa, we see that aflatoxin is the major mycotoxin. We see, depending on the different studies, you see that up to 100% of samples of poultry feed and feed ingredients is contaminated. And then if you look more specifically to the which rate or which concentration they are contaminated, you see like contamination levels up to 1 ppm, so 1,000 micrograms per kilogram. For example, this was in a study in Tanzania. And if you then know that the regulation is only like 5 to 20 micrograms per kilogram, I think then it's clearly put it into perspective that concentrations can be very high in your region. So for that's very important, but it's not only these very high levels of mycotoxins, which is can cause a negative effect on animal health. Today, we also talk about lower levels, even levels which are allowed by the European Union can still cause a negative effect on the animal health. The next major mycotoxin in your region is fumonisins. Fumonisins are depending a little bit on the climate. That's also something we will talk tomorrow on. Certain mycotoxins are produced by certain fungal species, and certain fungal species produce certain mycotoxins. And there's also some kind of crossover that a kind of fungus can produce different mycotoxins. So that actually mostly, if you look in the field, it's not just one mycotoxin, but it's a combination of different mycotoxins. If we look then to the other fusarium mycotoxins, besides fumonisins, deoxynivalenol, zaleron are also present in many cases, but the issue is that they are not monitored as frequently as aflatoxin is. But in a recent study, I've seen that for 2020, like deoxynivalenol is probably also the major contaminant for South Africa this year. So these are also very important. Let's discuss now the mode of action. So the mode of action means that how are these toxins causing toxicity? So as I mentioned, the toxicity is related to this chemical structure, and the chemical structure was quite diverse. So this mode of action is very diverse over the different mycotoxins. So aflatoxins, what it's doing actually, when it's present in the feed, it's kind of inactive. It still needs a kind of activation in the body before it's causing toxicity. And then we call that a kind of epoxidation, which happens in the liver. And then from that moment onwards, it can bind to DNA, RNA. So making it actually, first of all, very important for the liver. So it's causing a lot of liver toxicity, but very important, it's a carcinogen. So also for human health, this is a very important mycotoxin. So the next group of mycotoxins is deoxynivalenol T2 toxin. In contrast to aflatoxin, which actually needs this activation in the body, this mycotoxin doesn't need this activation. It's already active when it's in the feed, but that's not really binding to the DNA, but that's binding to the at stage of the ribosome, meaning actually it's binding to the proteins, more affecting on the protein level, not on the DNA level. There are some difference between, let's call deoxynivalenol and T2 toxin are kind of brothers, but T2 toxin is the more toxic brother of deoxynivalenol. The next toxin we're going to discuss, also some effects on today, is zaleron. This mycotoxin is even of more importance for pigs, for porcine health, if you compare it to chickens, because it's binding to the estrogen receptor. And this binding to the estrogen receptor actually is causing hyperestrogenism. But we see that there is a difference in the sensitivity for the receptors which are present in a pig compared to the one which are present in a chicken. So pigs are more sensitive compared to chickens. Fumonisins, it's also a potential carcinogen, and there it's affecting the sphingolipids. So actually it's the layer which composing the cell, which the cell consists out of these sphingolipids, and making these fumonisins attacking this layer, so making it toxic for the cells. And ochratoxin, it is the mode of action is quite diverse. It can affect in different ways, causing oxidative stress. So, but as you see, it's quite diverse. I don't want to remember you all these mechanisms, but just remember, okay, this toxicity is quite diverse. The mode of action is quite diverse. And when we talk about in 2 days about how can we mitigate this, of course, that also means that there will not be one single golden bullet solving every problem of mycotoxins. If we look to sub-Saharan Africa, mycotoxins, the concentrations, the contamination ratios, so the frequency of occurrence is much higher because, of course, it's hot climate. And you see sometimes even levels which negatively affect human health. But also animals are definitely at health. Also, not only production animals, but also many dogs are dying from aflatoxins in your region. But let's today talk about the impact of these mycotoxins on poultry health. And let's try to focus on the situation in sub-Saharan Africa. So if we look to the impact on animal health, we see it doesn't matter if we talk about the chicken, if we talk about the cow, if we talk about the pig, we see that the gastrointestinal tract is one of the major organs at risk, but also the liver, the heart, the lungs, the kidneys, the brain, the nerves. So many different organs are at risk. Some mycotoxins, mainly like ochratoxin, will mainly affect the kidneys. Aflatoxins will affect the liver. And many mycotoxins will affect the gastrointestinal tract. But also, besides the organs, it's also affecting the immune system. It's affecting the animals to resist against other infectious diseases. It can affect vaccines efficacy, and it can affect also breeding success. So many different organ systems are affected by these mycotoxins. So let's go more into detail. First of all, a general overview. Different mycotoxins affect poultry health in different way. You've got some causing, for example, this dioxin-1, T2 toxin, which we mentioned, can cause lesions, oral lesions, which we'll discuss. Some will more affect the rest of the gastrointestinal tract. Ochratoxin will cause some kidney problems. Some will decrease the egg production, fertility. And the last aspect I also want to make or demonstrate you today is that there can be some residue problems. So actually a food safety problem with products derived from chickens, eggs, meat, and liver. So if you have, let's call it a little bit higher levels of mycotoxins, you sometimes still see clinical effects. You sometimes still see really clinical symptoms, lesions. If you have got a little bit moderate, low levels of mycotoxins, in many cases, you will not see the effects, or you will not see these typical lesions. But keep in mind that it's not because you don't see a lesion that there is no problem of mycotoxin. But let's start with aflatoxins. Aflatoxins can affect animal performance. You can have a decreased feed intake, decreased body weight. Also, the egg production is decreased. And besides of that, it's affecting the immune system. It's decreasing the immune response. And sometimes you will also give you some pictures in the coming slides. You will see an effect on the carcass. So you'll see the pigmentation, which is not as it should be. And when you've got a little bit higher concentration, you'll definitely also see an increase in mortality. And if you then specifically look on necropsy to, okay, which kind of lesions can I then still see, is that the liver in the more acute phase, and that's what you mostly see on necropsy, is that liver is enlarged. It's pale yellowish because you've got an accumulation of fat there. Sometimes you might have bleedings, you have hemorrhages on the intestines, on the livers. And besides that, of course, I said, okay, it's inducing decrease of the immunity, so immune suppression. So also these organs are mostly a little bit small. You've got atrophy of the bursa, thymus, and the spleen. And I don't know if there are any people doing histopath. So if you do histology, you can see some necrosis of these hepatocytes, and then the next phase that there will be some lipid accumulation. But let's see some pictures, because pictures are always easier to understand. And here you clearly see that this is a liver of a chicken, which was fed a normal diet, and this is a liver of a chicken which was fed a contaminated diet with aflatoxins, about 1,000 micrograms per kilogram. And there you already see that this liver is enlarged. This is a pale liver because of this lipid accumulation. But on these pictures, you can even see this much better. Here you see these are livers from control group. So these are livers from chickens which did not receive aflatoxins. And here you see livers from chickens which received aflatoxin contaminated diet. And here it's even more higher concentration, where you see the liver is even more enlarged, even more yellowish. So clearly demonstrating these symptoms of lipid accumulation in the liver dysfunction. So that's really something typically for aflatoxins. Besides, okay, there can be a differential diagnosis, but if we talk about the mycotoxins, then we've got this type of liver. It's really typically for aflatoxins. In some cases, you may also have enlarged kidneys. Sometimes you more have these hemorrhages also there, but enlarged kidneys can also seen, for example, with ochratoxin. So that's not typically for aflatoxins. I also mentioned that the pigmentation of the carcass might be affected. Here you can see the more pale carcass, pale feet, when it's aflatoxin contaminated. Here, this is a normal chicken. So that can also be an indication of aflatoxins. The next mycotoxin we mentioned that's very important in your region is fumonisins. Fumonisins are actually mostly feed needs to be contaminated at really high level. If we compare aflatoxins with fumonisins, we are really talking about toxicity and regulation in Europe of like 20 micrograms per kilogram. If we talk about fumonisins, we are talking about a range of 20,000 micrograms per kilogram feed. So meaning that fumonisins are not as toxic as aflatoxins. Also, you don't have this really typical characteristic clinical symptoms when the feed is contaminated. In contrast with pigs, in pigs you might sometimes have these pulmonary edema, so respiratory symptoms. So fumonisins in poultry mostly just giving decreased performance. And also lesions, it's mostly sometimes just enlarged organs like a kidney, a liver. So not that clear symptoms, even if you've got very high levels. Histopathology, so you can have some liver necrosis with some biliary hyperplasia, but also again, this is not pathognomonic. Trichotocin, so deoxynivalenol T2 toxin, is much more toxic compared to fumonisins. This also gives performance results, decreased performance result, decreased feed intake, sometimes even feed refusal. Deoxynivalenol might also be known by some of you under the name of vomitoxin, because this mycotoxin is inducing vomiting, but not in chickens, but in pigs. So pigs are more sensitive even for that symptom. It's sometimes give an abnormal feathering. I will demonstrate you, will show you a picture, but I have to admit that this is a symptom which I've never seen in practice. So, which is very important. That can also negatively affect the gastrointestinal tract, which might lead to a kind of dysbiosis status and also induce diarrhea. In some rare cases, it might be a bloody diarrhea, so but diarrhea. And that really characteristic for higher concentrations of deoxynivalenol, but definitely of T2 toxin, is necrosis of the oral mucosa, and then giving the characteristic black tongue. And the last one, gizzard erosions. So on necropsy, you might see this necrosis in the beak, this oral mucosa. You can also sometimes see this congested redness of this intestinal mucosa. Sometimes you see some mottling on the liver, atrophy of the spleen. But let's look to the pictures. So it's mainly, this mycotoxin is mainly something which is affecting the gastrointestinal tract. And here you clearly see this, you see necrosis of the tongue. So there is also the reason why it's called black tongue disease, only the tip. Here you see more necrosis, and also here. That's definitely something which might still be seen frequently in practice. Of course, take into account that also other factors can induce this black tongue when there is, for example, something wrong if you use acids. But this is really kind of burn reaction. It's a direct reaction, also showing that this mycotoxin doesn't need a biotransform. It doesn't need to be activated in the body. This really demonstrates it's really immediately toxic. Besides that, of course, the next aspect, which is typically, maybe this on this picture is not that clear to demonstrate, but here you see the beginning of gizzard erosions. This is much more clear, this picture. Here you see clearly these gizzard erosions caused by these type of mycotoxins. And on these pictures, this reddening of the gastrointestinal tract, mainly these are, as I mentioned, these symptoms you only see mostly when you've got very high concentration. And again, it's not because you don't see these symptoms that there cannot be a problem with mycotoxins. And this is a picture taken from a book, because I never have seen this because of the effect of deoxynival or T2 toxin is some feather abnormalities, but it's described that it can occur. Another mycotoxin, ochratoxin A. So it can also give more general symptoms, but this is a nephrotoxic, so it's toxic for the kidney. So we have, of course, as a symptom that water consumption might increase, that the litter quality will, of course, as a result, not be that optimal. And it's, for the rest, it's mainly performance. You will characteristically, we will see these pale, swollen kidneys. And as a secondary effect, because your kidney function is not working as it should be, you can have some coat. We will see in the next slides. And sometimes you have some effect on the liver. So here, characteristic is swollen kidneys is typically something which you can see with ochratoxicosis. As I mentioned already in the introduction, Zaleron is actually much more toxic for pigs than it is for poultry, but also in poultry it's described that you can have a negative effect on fertility. And this was the mycotoxin which was binding to this estrogen receptor. Also, also the lesions are associated to that. You can have hypertrophy of the oviduct. In some cases, mainly in turkeys, it's described that can give some cloacal swelling, and the sperm quality might also be negatively affected. In some cases, there are some testis atrophy described, but let's say in most cases, Zaleron will not induce clinical symptoms. But that doesn't mean that it cannot have a negative effect, but mostly not. We will not see any clinical symptoms. Now I described you which kind of clinical symptoms, which kind of lesions you might detect on necropsy, some cases, some histopathology, but is that what we always see in the field? In some cases. In some cases, yes, you will have clinical symptoms, but in many cases, you will have a negative effect of mycotoxins on animal health, but you will not see clinical symptoms. Because in many cases, we are talking about lower levels, moderate levels of these toxins, and this will affect the immune system, will affect the gastrointestinal health, also other types of processes, physiological processes, but it's much more difficult to diagnose. And tomorrow we also talk a little bit about how you can diagnose mycotoxins. But in the end, it ends the question, okay, is there a one-on-one situation? If I have a certain effect, a certain concentration of mycotoxin, can you then predict what the effect will be on animal health? No, I cannot predict, because the outcome will depend on which mycotoxin, what's the concentration, how long are the animals exposed. And as I mentioned, that mycotoxins are the same mycotoxin can produce by different fungal species, different fungi, but the fungus can also produce different mycotoxins. So in the field, also we combine different feed ingredients. It's mostly a combination. It's not only aflatoxins, but it's aflatoxins and fumonisin. It's aflatoxins together with the zeluron, with fumonisin maybe. And this cocktail will also have an effect. It's also not easy to predict what the effect there will be. You also see the different poultry species that turkeys are a little bit more sensitive than, for example, layers. Broiler chicks, or in general, young animals are more susceptible for the effects than older animals. If the management factors, for example, if you've got a good vaccination schedule, then sometimes maybe lower moderate levels might also not be that big issue. But if you've got already problems with the immune system, with your vaccination is not optimal, then this exposure to mycotoxins might just be, or might just help or might just make the animal more sensitive for certain infectious diseases. This is just from a review paper demonstrating that many different mycotoxins have a negative effect on different aspects. So nutrient digestibility, different enzymes, very important, the intestinal barrier system, immunity. So many different aspects of the intestinal health are affected by mycotoxins. So actually by that, they make the animals more prone to have some gastrointestinal problems. So for example, aflatoxins, negative effect on the intestinal permeability, even low concentrations. So what does this mean? Intestinal permeability is these cells in the intestine are connected to each other. We call it with like disjunctions, and they are connected, they regulate which ingredient, which particle, which food is going through, which pathogens are. So actually, they really regulate feed intake. So it's going from the gastrointestinal tract to the bloodstream. And from the other, okay, they say, no, this is a pathogen. But what's now doing, these mycotoxins are doing actually just opening. It's kind of opening the door. For example, there are studies demonstrating that E. coli can easily pass them. The gastrointestinal tract is regenerating very frequently. So when there's kind of damage, in just a few days, everything is healed. But that's also affected by mycotoxins. So the healing process after that is kind of damage. It's not going as it should. The mucus layer is actually the first barrier of the intestinal tract. So mycotoxins affect that. Immune response is certain immunoglobulins, cytokines, all immune response. We see also an effect on the types of bacteria, good bacteria, the intestinal microbiota, which are present, and it's causing oxidative stress. So this slide clearly demonstrates that many different mycotoxins have a negative effect already at the gastrointestinal tract without any lesions that you will observe. So, but you have to take that into account because that's of course affecting performances. So this negative effect on the gastrointestinal tract will affect how the chicken will absorb nutrients, will affect how it can absorb other toxins, but also veterinary drugs. And because they have to also pass the same barrier. And sometimes we even see that there is an interaction with mycotoxins and veterinary drugs. But it can be that, for example, you have therapy failure induced by mycotoxins because the drug you give is not absorbed as it should be, for example. And we will demonstrate also that it's making animal more susceptible for infectious diseases. So, but let's start with the beginning. What's the effect on growth? Yeah. We see that mycotoxins can have an effect on feed intake and have an effect on body weight gain. So your feed conversion will not be as it should be. For example, in a study in Nigeria, we see that when the feed was contaminated with aflatoxins up to a concentration of 1,000 micrograms per kilogram, you clearly had a negative effect on these performance parameters. But if you even then give lower levels, for example, 500, there was also a study with 500 micrograms per kilogram, also have a clearly negative effect on body weight gain. And then when I went through all studies published in literature, you could even see when there was concentration only like 50 micrograms or 100 micrograms in layers already have a negative effect on performances. This study, I think clearly, or this picture really clearly demonstrates, so the animal on the left was fed mycotoxin and aflatoxin contaminated diet at 0.5 ppm. So this means 500 micrograms per kilogram. And this was for the control diet. So clearly affecting this performance. If we look to layers, we see that layers or breeders, you see that certain mycotoxins like aflatoxins have negative effect on the production. So how the amount of eggs you will get, but also on the quality. You will see that the eggshell quality can be affected by mycotoxins, or also how big your eggs are, so the egg weight. Also for fertility can be an issue. And there was a study that also demonstrated that some mycotoxins will increase the number of rejection because of course, this poor eggshell quality. In this study, they used really a high amount of aflatoxins. As I said, this is really high. But in that case, they had even 1 on 3 eggs was not as it should be. As I mentioned already in the introduction, also something very important is food safety. Although I think we can be in general be lucky with our mycotoxins in poultry, that the level which is transferred is quite low. But there is a risk, and mainly the risk is there for aflatoxins. But if we compare it, for example, to the dairy sector, there we see that aflatoxins are transferred much more from the cow, from the blood from the cow to the milk. So levels are much higher in milk, but also in eggs, in meat, in liver, we can have some concentrations of aflatoxin. So what's happening? The feed you produce it on your farm, feed you purchase. So both it's not because you purchase it or that it will not be contaminated. So both is an option. So you feed it and then you will see some transfer of the mycotoxin. If you feed a diet which contains like 800 to 1000 micrograms per kilogram, so ppb, see, you only end up with concentrations of 1 to 2 ppb. But if you take into account that this mycotoxin is a carcinogen, you really don't want this in your food. So therefore, it's also very important that you not only for the health of your chickens, but also for your own health, that you don't feed aflatoxin contaminated feed to your chickens. In a study in Mozambique, almost 40% of livers of chicken livers were contaminated with aflatoxins, concentrations up to 2 micrograms per kilogram. And in a study in Cameroon, actually almost half of the eggs that they sampled in this study contained some levels of aflatoxin. So, and these levels were even quite high, 7.4 micrograms per kilogram, because levels which are allowed for food are, of course, even much lower compared to the levels which are allowed for feed. So this mainly like chicken eggs can be definitely, if you feed quite high levels to your chickens, remember that it can be important for your food safety also. A few words on vaccine efficacy. I mentioned already that in case you have good vaccine schedule, okay, you cannot vaccinate against mycotoxins. That doesn't exist at the moment. But if you can protect your animals against many other diseases, then it might also help, because if you have a diet which is contaminated with mycotoxins, you will see that animals are more sensitive for many diseases. But from the other hand, also, you will see that mycotoxins can have a negative effect on your effect of your vaccine. In some studies, mainly done with Newcastle vaccination, so you clearly see when a diet was contaminated with aflatoxin, you had a negative effect on the immune response. So in this graph, you see after weeks after vaccination, and this is about the titer. So the titer you can see for it was for Newcastle. So a normal diet, so the full black line. But when the diet was contaminated with aflatoxins, you clearly see lower titers in the blood. And it was almost comparable with IBDV, so with Bristol disease virus. So really something which is immunosuppressive. It's also this aflatoxin is immunosuppressive. So you think in that case, you vaccinate your animal and you think, yeah, okay, I'm protected now. That might not be the case. Although I have to say that there at the moment are not really studies available where they first vaccinate the animals when the diet is contaminated and then really try to infect them. This kind of studies are not available, but demonstrating that you've got a lower immune response after vaccination, they are definitely. This one is an example, but also, for example, other studies where they used an attenuated live vaccine for Newcastle, infectious bronchitis, and Gumboro. Vaccinate layer chicks for all three diseases. They had lower antibody titers. Another study, they vaccinated adult layers against Newcastle, infectious bronchitis, and Gumboro in that study, and same. So not only in young animals, but already also in older animals. And remarkably, the concentrations were only like 200 micrograms per kilogram. So actually, these are concentrations which are frequently observed in your region. Mycotoxin contamination affects the gastrointestinal tract at many different levels. So at the gastrointestinal tract, animals will have a damaged gut, so they will be more sensitive to, for example, coccidiosis. Coccidiosis and Fusarium mycotoxins. I don't think there are any studies done with aflatoxins, but it has been demonstrated when the feed was contaminated with different Fusarium mycotoxins, so deoxynivalenol, zearalenone, combination with fumonisins, the negative effect on the intestinal immune response against coccidiosis. Also on the regeneration after infection with coccidiosis. And this study, it's even more interesting than the previous one. You also clearly see that the severe, because here, at the y-axis, you see the lesion scoring of the duodenum, jejunum, and cecum. You clearly see an increase in the lesion scoring when the diet was contaminated with dioxynivalenol or fumonisins or the combination. And this was mainly observed in jejunum and cecum. So it seems to be that the effect is also depending on the section of the intestine, or there might be also depending on which Emeria species. So clear for Emeria maxima and Emeria tenella, the lesions were more severe when the diet was contaminated with mycotoxins. And then during my PhD, I've been looking to the interaction between mycotoxins, fumonisin, and deoxynivalenol, and necrotic enteritis. So necrotic enteritis is caused by Clostridium, and this is an opportunistic pathogen. It's actually really needs nutrients in the gastrointestinal tract to start proliferating. So what to see that mycotoxins are affecting the gastrointestinal tract of many different parts. So it's negatively affecting viability, proliferation, integrity, so as the one I mentioned, the permeability, mucus layers, microbiota, oxidative stress. So actually, these mycotoxins created or creating a gastrointestinal tract, which is a perfect environment for the bacterium, because there are many nutrients now coming available. Because, for example, when you've got a dead enterocyte, so a dead cell, this can be used by bacteria. You've got protein leaking from the blood to the gastrointestinal tract. So this was really a perfect environment for the bacterium to start proliferating. And when this bacterium is proliferating in the gastrointestinal tract, then it also can start causing disease. So there we had more chickens with necrotic enteritis. So it's not that mycotoxins are the cause of necrotic enteritis, they just actually make it easier for the pathogen to cause the disease. So we did some infectious trials where we artificially infected our chickens with Clostridium perfringens. We either did not inoculate the bacterium, but if we inoculated the bacterium and the diet did not contain mycotoxins, we observed about 20% of chickens with the disease, with necrotic enteritis lesions. When the diet was contaminated with deoxynivalenol, we observed like about 50% of chickens with this disease, with necrotic enteritis. And this 3 to 4 milligrams per kilogram feed is actually even below European guidelines level. As I mentioned already, it's not this. You don't see clinical symptoms of deoxynivalenol at this level, but it's making the animal more susceptible for this disease. So we call them a predisposing factor. We did the same for fumonisin, and also here we see that the number of chickens with necrotic enteritis increased when the diet was contaminated with fumonisin. So what I want you to remember from the presentation of today is that we have seen different mycotoxins. They all have a different mode of action. And you see that different organ systems are affected. And you see remember like the gizzard erosions you might have. You might have these necrosis of the oral mucosa from the trichotillomyces, this really large pale livers, which is seen with aflatoxins, these swollen kidneys, which we might have of ochratoxins. These kind of lesions and also the associated symptoms you mostly only see with high contamination levels. It's important to look to that. But also lower levels, moderate levels of these mycotoxins will have a negative effect on poultry production, on performances. Sometimes you don't see the symptoms, but you still have negative effect on performance or on immunity. Also remember that, okay, although the risk is lower, but there is still some food safety risk. And mainly this is for aflatoxins, because there can be transfer from aflatoxins to eggs, to liver and meat. And since this mycotoxin is a carcinogen, we don't want that, of course, in our food. So keep that in mind. Mycotoxin exposure can also induce vaccine failure. Although you vaccinate and you have a good vaccine schedule, if the diet is contaminated, maybe your effect of vaccination might not as you would like to have it. So it might not. So actually, also it's an investment. Vaccinating is an investment. So therefore, also it's important to have a look to these mycotoxins to make sure that this is a good investment and that you have an optimal return on investment. Lower levels of mycotoxins might have a negative effect on all different aspects of gut health, of the intestinal barrier function, making that the absorption of nutrients is not optimal. You might have some interference with other toxins, with antimicrobials even. Make the animal more susceptible for infectious diseases. Remember, for example, necrotic enteritis, coccidiosis, and in general, mycotoxins might affect the immune response and infectious diseases I mentioned already. And with that, I'm at the end of the presentation of today, and I would say I'm open for questions.
Hi, Gunther. Thanks for your nice presentation.
Okay, so the first question, which kind of mycotoxins affect poultry and humans at the same time? Actually, many of them. For example, aflatoxin, I think for your region is definitely a good example. You have many negative effects in poultry, and it's also a very big problem for human health because it's a carcinogen, but also many things we have demonstrated, like the effect on the gastrointestinal tract that we see for many different mycotoxins. It's also observed in humans, for example, deoxynivalenol. So most mycotoxins I presented today can also negatively affect human health. But okay, if we then look about food safety, it's mainly a problem of aflatoxins. The rest is really at very low level in poultry products. But since, of course, we don't eat only meat, we also consume cereals, for example. We drink a beer, and even beer can be contaminated with mycotoxins. Wine can be contaminated with mycotoxins, like ugali, for example, in your region. There you have many, definitely aflatoxins can be in there. So it's not one single mycotoxin. The next question, is there any possible prophylactic remedy? Well, in the third webinar of this week, we will really focus on that topic, and we will tell a little bit more about how we can have interventions at field, because the mycotoxins, some of them are produced on the field. Some of them are actually more produced during storage. For example, aflatoxin is more a storage mycotoxin, so the levels will increase during storage when you don't store your cereals as you should, for example, your corn. And then you can do some interventions over there. But also when you just receive your feed, you still can do some post-harvest mitigation strategies. For example, adding some mycotoxin binders or detoxifiers might be one of the options you can do. Well, patulin, that's a very good question, because patulin is, how does patulin as a mycotoxin can affect poultry? Patulin is probably one of the upcoming mycotoxins, let's call it. So one of the newer ones, maybe. We see that if we talk about global warming, you see that currently aflatoxins is really the number one mycotoxin in Africa. But it's expected that patulin will be one of the major ones for the future, because the fungus which is producing patulin is really a fungus which which is producing mycotoxins up to very high temperature. So if the environmental temperature is increasing in the future, probably we will see even more patulin. But then coming back to the question, impact on poultry health, let's say that's still a big question mark. There are not many studies really looking to the effect of patulin on poultry health. But what I see in other animals, there will definitely be an effect on like performance. but no, it's fair. The number of questions is very limited.
So does cooking or any other kind of food processing activities on eggs, on meat or meat, can lower the toxicity to humans?
Yeah. Yes, there are, and it's also something I will definitely explain a little bit more on Thursday. There are some food processing and cooking techniques which can help, but not just cooking itself. It doesn't work. Temperatures are not even high temperatures. Doesn't matter for mycotoxins. But what you like, for example, what is done like sorting of the cereals, sorting of the grains, that definitely lowers the level of mycotoxins. Cooking techniques for alkaline cooking, it's something which is more described in Latin America, that can lower the level of fumonisins, for example, like described for tortillas. Then there are recently we also did some work together with the University of Johannesburg on fermentation. Some fermentation techniques might also decrease some mycotoxin levels. But we will talk a little bit more even on Thursday on that.
Okay, I think, yeah, you have here the other one. So this was the first one. Can acute mycotoxicosis cause paralysis?
Yeah, sometimes it's described with aflatoxins. That can also give some nervous symptoms. But
Okay.
I would also look to other reasons, because that's not the number one, but it can occur with aflatoxins.
Okay. Is there a technique that detoxify mycotoxins in the gastrointestinal tract of animals? If there's anything that we can do?
Yeah, the same answer. I would say that's a perfect introduction for the webinar of Thursday. There we will talk much more about mycotoxin binders, detoxifiers, and how they work.
Yeah, okay.
Does mycotoxins
mycotoxins that is in low concentration in animals can show clinical signs in humans?
Clinical signs in humans, there are, of course, humans are all more sensitive for some of mycotoxins than chickens. And there is also an important aspect, age. As I mentioned, the age of the animal is important, but also the age of us, of course, is important. Like, for example, I think one of the best or well-known symptoms is stunting in children. But even deaths are every year, there are some deaths all because of aflatoxicosis. But looking to the metabolism, we as a human are more comparable as for sensitivity with a pig than with a chicken.
Okay. Okay. So next one: can the mycotoxins spread amongst flocks? Are they contagious to humans?
It's not an infectious disease, so it's really a toxin. So the toxin should be present. Toxin should be present in the feed. Although in the dust, probably if you will try to detect them in the dust, you will probably also find some. But it's really the feed is the source of exposure. So if you're using the same feed for the different flocks, you might have problems in a different flock. But it's not like a virus which can spread by the air. No. And being contagious for humans, then we talk again just about the food safety aspect. So it's not something which is spreading like that in the air. But I have to say one but on that, and an important remark is people working in feed mills, feed factories, because they work a lot with the feed, and then in the dust, something it's already this. Some, I think, researchers from Spain mainly worked on that. That high levels can be detected in the dust in feed mills. So from an aspect of safety, their wearing masks is very important for that. But it's not like a virus to spread.
Yeah. Okay, so the last one, I think, what are the other clinical signs of mycotoxins different from coccidiosis as the major cause and not being microbial contamination? Yeah, I didn't have time to rearrange this one, but I think the question here is, what are the other clinical signs that you can see besides coccidiosis?
Well, yeah, as I mentioned, coccidiosis it's not it's just making animals more susceptible for coccidiosis.
Yeah.
It's not really a clinical sign of the toxin itself. It's just a clinical sign of the immunosuppression. So
Yeah.
of the clinical symptoms are, if you see them, is this enlarged pale liver for aflatoxins, these gizzard erosions, these black tongues for the trichotillins, these enlarged kidneys for ochratoxin. That are really the clinical symptoms. The rest is just a result of the immunosuppression, of the negative effect on the gastrointestinal tract. It's just a result of it. It's not a clinical sign of it.
So thank you, Gunther, very much for your presentation. It was very nice. I don't know, Eric, if you want to say anything before closing up.
Yeah, just I want to thank you, Professor. I want to thank for this excellent presentation. I think it was a very nice introduction webinar, and for the next one tomorrow, you will dive into analysis and monitoring, how to detect it, how to confirm it, and then how to monitor it in feed. And then the day after tomorrow, you dive into feed additives and detoxification. Thank you very much. There was a lot of questions, I think, but apparently it's not a problem for you to answer them. That's very nice to hear. And thank you very much, Tiago, Hilde, Aurelie, the people from Vetworks. You're making this this webinar a real success. I hope to see all of you tomorrow. Thank you for participating. I'm very happy with the number of people who showed up, so I hope to see you again tomorrow. Okay, thank you.
Thank you. Thank you, everyone.